The human genome contains an estimated 25,000 genes, all of which can have variations leading to genetic conditions. Until now, understanding many of these conditions has been difficult, as access to genetic data has been restricted. But a huge dataset from more than 60,000 people has been made freely available, and has already had a major impact on clinical research and diagnosis of genetic diseases. The Exome Aggregation Consortium is a project led by scientists at the Broad Institute of MIT and Harvard. The genetic data from 60,706 people from diverse ethnic backgrounds, containing over 10 million DNA variants, has been made freely available. A new study has analyzed the data to show that genetic variation, which was previously undetected in small datasets, can now be seen in the XAC set. The findings apply in particular to so-called Mendelian diseases, caused by a single gene. Well-known examples include cystic fibrosis, which inflicts severe damage on the lungs and the digestive system, Pfeiffer syndrome, characterized by a severe deformation of skull bones, and Smith-Lemli-Opitz syndrome, a disorder linked to multiple malformations and intellectual disability. At the same time, the researchers found that nearly 200 variants previously fingered as the cause of severe disorders appeared way too frequently to be a culprit. Daniel MacArthur, senior author of the study, said, "The success of XAC was made possible by the willingness of our colleagues in many large, disease-focused consortia to openly share sequencing data. The scale and diversity of the XAC resource is invaluable. It gives us the ability to discover extremely rare variants and offers an unparalleled window into the roots of rare genetic diseases." The genetic data was collected from tens of thousands of human samples, from researchers around the globe. The samples were then analyzed by a computer, to produce a catalog of human genetic variation in previously unseen resolution. Many of these variants had never been reported and are extremely rare, occurring in less than 1 in 10,000 people. Doctors can use the data to compare to their patients' genome sequences, to shed light on the genes that may underlie the patient's condition. If a variant found in the patient's DNA sequence is very rare in XAC, it then becomes a key suspect in causing the rare disease. The database was released in October 2014, and since then has had more than 5 million page views. It has also allowed doctors to provide more accurate genetic diagnoses for thousands of rare disease patients. Heidi Rem, medical director at the Broad's Clinical Research Sequencing Platform, said, "The XAC resource gives us incredible insight when evaluating a patient's genome sequence in the clinic. Because data until now has been limited, many DNA variants are rare or understudied. This has made it unclear if they have any effect on disease risk, and whether they should be taken into consideration when diagnosing and treating patients. Researchers can look at how often a variant occurs in the XAC database to rule out those that are relatively common, and focus on the true disease-causing variants." XAC has also been used by researchers to identify new rare genetic disorders. Matthew Hurls, a researcher at the Wellcome Trust Sanger Institute, said, "In our own research, using the XAC resource has allowed us to apply novel statistical methods to identify several new severe developmental disorders. Resources such as XAC exemplify the benefits that can be achieved for families coping with rare genetic diseases." The XAC dataset also revealed more than 100 previously reported disease-causing mutations to actually be unharmful. This reduces the number of false positive findings in databases widely used by clinical labs. Jose Flores, an associate professor at Harvard Medical School, said, "With its large sample size and high resolution across many populations, the XAC database provides much greater power to interpret rare disease-causing variants than ever before, even for common diseases."